Let me start with a simple observation. Have you noticed how siblings look alike but never exactly the same? One may inherit dad's nose, another mom's smile, but each child is unique. Why does this happen? The answer is hidden in a fascinating process called meiosis. Let's learn about it. Our body cells, that is, somatic cells, always divide by mitosis. This makes the identical copies of cells for growth and repair. But the germ cells, that is, cells producing sperm in male and eggs in female, divide by meiosis. This reduces chromosome number and creates variation. In humans, body cells have 46 chromosomes, that is, they are present in 23 pair, that is, 2n. We can call it as diploid. But in sperm or egg cells, there are total 23 chromosomes each, and they are present in haploid form, that is only n. So after fertilization, sperm cell plus egg cells, that is each with 23 chromosomes, produces the zygote with 46 chromosomes, which is same as your body cells. So meiosis is the reason siblings don't look like photocopies. Instead, each of us is a limited edition remix of DNA. Now let's see some key terms before learning the meiosis. Homologous chromosomes, which means a pair, one from mother and one from father. They are same in size, shape and gene loci, but alleles may differ. For example, both carry a gene for eye color. From mom it may be brown and from dad it may be blue, but allele is for eye color. Like a pair of shoes, same design, left and right slightly different. So these are called homologous pair. Next is synapsis, pairing of homologous chromosome during the zygotene of prophase I. This process is called as synapsis. Chromosomes are held together by the synaptonemal complex, that is like a zipper. Just think about it like two dance partners holding hands together to start their performance. Next is chiasmata. It is like X-shaped points where chromosomes remain joined after crossing over. This formation takes place in the Pachytene stage of prophase I and it is visible in Diplotene stage. Think of it like two friends who swapped their caps and are still holding the hands before leaving. That is the exchange of genes. Here is one more thing I want you all to understand: the chromosome numbers. So we will count chromosome by the number of centromere present. So this is also one chromosome, and after duplication in S phase of the interphase, this is also one chromosome, but two chromatids are present attached to the single centromere. So these are 46 chromosomes with 46 chromatids, and these are also 46 chromosomes but with 92 chromatids. This concept is very important in the meiosis I, where homologous chromosomes separates but sister chromatids stays together. Now let's learn the main story, that is the meiosis. It happens in two steps: meiosis I, which is called as reductional division, and meiosis II, which is called as the equational division. As the mitosis, it also have the phases like prophase, metaphase, anaphase and telophase. Now let's see the meiosis I, which is also called as the reductional division, where diploid cells divide to form the two haploid cells. So first is prophase I, which is the longest and most important with 5 subphases, that is leptotene, zygotene, pachytene, diplotene and diakinesis. I will give you one mnemonic to remember these phases later. Okay, so in first step, that is leptotene, chromosomes appears as thin straits. So here chromosomes are preparing to get condensed. Next is zygotene, where homologous chromosomes pairs with each other. Homologous chromosomes are the chromosomes which carry the same alleles, or you can say which are same in characters. And synapsis is formed in this phase. Synapsis is like the zipper-like structure, that is two chromosomes holding their hands. Next is Pachytene. It is the most important phase where the crossing over of chromosomes occurs, that is the exchange of genes occurs. So in this stage, chromosomes swap their characters. That's how the variation created. Next is Diplotene. So after sharing the genes or characters, synaptonemal complex breaks down and chiasmata is formed. So chiasmata is like chromosomes are still holding their hands and that X-shaped marker is the proof of crossing over. Next is Diakinesis. Our chromosomes are fully condensed and ready for next phase. Nuclear envelope disappears and spindle starts to form. So in Prophase I, remember these three main important phases. Zygotene, where synapsis is formed, that is the handshake. Next, Pachytene, where crossing over occurs, that is the exchange of genes. And Diplotene, where Chiasmata is formed, that is the X shape formed by the crossing over of chromosomes. Now next is metaphase. The homologous pairs align at the equator randomly. Remember, in mitosis, chromosomes align at the equator, but in meiosis, homologous pairs. So here we use pairs. Those 46 homologous pairs align at the equator randomly. It's like shuffling a deck of cards. Each shuffle and new combination is. So how will you remember? M for middle of the cell. Next is Anaphase I, where homologous chromosomes, that is the pair of those homologous chromosomes, separates and go to the opposite poles. So here the breakdown of pairs takes place, but sister chromatids stay together. And last is Telophase I. Two haploid cells are formed, but chromosomes still in the duplicated form. So what is the result of meiosis I? The two haploid cells are formed from one diploid cell. So diploid cell having 46 chromosomes but in duplicated form. Haploid cells having 23 chromosomes also in the duplicated form. So here is the number of chromosomes and number of chromatids matter. Okay, now let's move to the meiosis II, which is also called as the equational division, and it is same like the process of mitosis, having prophase, metaphase, anaphase and telophase, but named as the II. In Prophase II, again chromosome condenses and spindle reforms. Next is Metaphase II. Those chromosomes which are present in duplicated form align at the middle of the cell. So metaphase, M for middle. Next is Anaphase II. The sister chromatids separate. So here chromatids separates. But in meiosis I, chromosomes are separated. So sister chromatid separates and pulled apart. Anaphase, A for apart. And next is Telophase II, where 4 haploid gametes are formed. Why 4? In meiosis, 2 haploids are formed from one cell. And from those 2 haploids, here again 2 haploids from each haploid cell formed. So total there are 4 haploid gametes. So final result is 1 diploid cell to 4 haploid gametes, all genetically unique. Why? Because in Prophase I there is the crossing over and exchange of genes. So here completes the process of meiosis. So here are some mnemonics to remember the stages of meiosis. First is pass me a teacup, that is prophase, metaphase, anaphase, telophase, and last is cytokinesis. And to remember the Prophase I subphases, Leptotene, Zygotene, Pachytene, Diplotene and Diakinesis, use the mnemonic little zebra pens, dirty drawings. Okay, I hope this may help you. Now let's see the significance of meiosis. So why does meiosis matter so much? The very important reason is variation. In meiosis I, where crossing over takes place and independent assortment happens, the unique gametes are formed. So we humans are not copy-paste files, but we are the limited edition. Next, to maintain the chromosome number. So during the fertilization, when haploid sperm cells having 23 chromosomes and haploid egg cells, which is also having 23 chromosomes, will combine to form the diploid zygote, which will have 46 chromosomes. That is the diploid state. So the chromosome number is maintained as the body cells. Next is sexual reproduction. Meiosis is very important process in the gamete formation. Next, meiosis plays evolutionary role, that is variation, adaptation and survival. Here are some NEET key points for my 11th and 12th grade students. Prophase I, which is the longest and most important. Remember the substages. Little zebra pens, dirty drawings, where synapsis is formed in the zygotene, that is the zipper-like structure. Next is crossing over formed in the pachytene, where genes are exchanged, and chiasmata, which is formed in the diplotene, that X-shaped structure after the crossing over. Also remember, meiosis I is the reductional division, where diploid cell divides to form two haploid cells, and meiosis II is the equational division. Those two haploid equally divided to form four new haploid cells. Here is one more thing which is also important, that is the non-disjunction disorders, which are formed by the failure of chromosomes to separate properly during the meiosis process, which may cause trisomy or monosomy. The examples of these disorders are Down syndrome, which is also called as trisomy 21, and Turner syndrome, where female is only with one X chromosome, and Klinefelter syndrome, where male is born with extra X chromosome, which may result in developmental, physical, and fertility issues depending on the disorder. So, here is the summary of meiosis, which occurs in two stages: meiosis I, which is called as reductional division, and meiosis II, which is called as equational division. In meiosis I, prophase I is very important, which is subdivided into 5 subphases: leptotene, zygotene, pachytene, diplotene, and diakinesis. So, to wrap it up, meiosis is the life's secret recipe for uniqueness. Without it, every child would be a Xerox copy. Thanks to meiosis, each of us is a limited edition masterpiece, carrying traits of the both parents, yet entirely original. Revise mnemonics before NEET. Pay extra attention to the Prophase I and practice past year questions on meiosis and chromosomal disorders. If this lecture helped you, hit like, share and subscribe. Just like meiosis adds variation, your support adds energy to our channel. Thank you guys.